Good evening, everyone. I hope you can hear me loud and clear in the room, and a very warm welcome to tonight's discussion on genomics, presented by Peter Joshi, who we'll get to later. Now, as I say, we're physically here in Edinburgh tonight. We're also live streaming to, I think, over 100 people have signed up to this evening's event, so a very warm welcome to those people too. My name's John Taylor. I'm President-elect for the Institute and Faculty of Actuaries, and I will be chairing this evening's talk. So the topic for this evening is about genetics and the potential, genomics rather, than the potential impact this could have on actuarial work. Now, I think the potential for genomics to have quite an impact on actuarial work has been known for some considerable time, and indeed concerns around some of the negative impacts led to the ABI announcing a moratorium in 2001, where they said that anyone applying for life assurance need not reveal whether they'd had a genetic test or indeed the results. I think there was one exception to that, but that was the broad thrust of it. Now, the impact of that moratorium was rather limited at the time, I would say, partly because the understanding of genetics and longevity was somewhat limited, and indeed the availability of genetic tests was somewhat limited. Fast forward 15 years or more, and the impact that technology has had, both information technology and biotechnology, on both those issues has been pretty profound. So the understanding now between the relationship of genetics and longevity is much more advanced than it has been, largely in part to the work of researchers like Peter and his colleagues. And indeed the availability of DNA tests is changed beyond recognition. Even there are TV adverts by companies like 23andMe offering to do a health assessment based on your DNA of the order of £100. So we're in a radically different world, and I think we're in for a very interesting session this evening to explore the impact of genetics on longevity, on actuarial work, and indeed on some broader questions for society in general. So to start this evening, it gives me a great pleasure to welcome our opener, Angus MacDonald from Heritability University. Angus graduated in mathematics from Glasgow University and then joined Scottish Amicable Life Assurance Society, qualifying as a Fellow of the Faculty of Actuaries in 1984. In 1989, he then moved to Heritability University, obtaining a PhD in 1995, and being appointed professor only 5 years later, and serving as head of Department of Actuarial Maths and Statistics from 2007 until 2013. He was a member of Faculty Council from 1998 to 2007, and in 1999 he set up the Genetics and Insurance Research Centre, which has since produced most of the actuarial research on the subject, including 2 papers that won the David Garrick Halmstead Prize in 2005. Angus was elected Fellow of the Royal Society of Edinburgh in 2006 and was awarded the Finlayson Medal by the profession in 2011. He is currently Editor-in-Chief of the Annals of Actuarial Science. So a very warm welcome to Angus.
Right, well, thank you, John, and good evening, ladies and gentlemen. It's now about 25 years since genetic testing first came to the attention of actuaries in the mid-1990s, and in a very specific context at that time of mutations in single genes that were generally associated with severe disorder or extremely premature death. And had a clear pattern of Mendelian inheritance, which meant the pattern of inheritance was clearly evident in the families. So these diseases had been studied for a very long time. The only difference that the test made was that it turned a 50% chance of having inherited the mutation into knowing certain knowledge one way or the other. It led to immense controversy in the UK and in other countries. In particular, one of the most contentious points was the insurers' use of genetic tests. On the one hand, the people feared genetic discrimination and loss of privacy. On the other hand, insurers feared adverse selection. And this was dealt with in different ways in different countries, but in my opinion, in the UK, it was dealt with in a rather smart way by the Association of British Insurers, eventually. They got good genetics advice from the Professor Sandy Rayburn, who sadly died last year, and they entered into the Moratorium, which John mentioned. In its first incarnation was from 1996, and it's evolved since then, but it's with us still, and it's been very successful in taking genetics away from the headlines in the UK. There are a couple of points I would emphasize. First, an important feature of the moratorium is, and I think this is where getting good genetic advice was vital, was that it was restricted to predictive tests only, and it did not cover diagnostic and prognostic tests, tests which might in most circumstances be associated with a pre-existing condition. That's not always been the case. The most recent example of government's interest in the subject is in Canada, where Bill S201 was passed in May 2017 and became the Genetic Non-Discrimination Act, and that is completely sweeping. Insurers may not have access to any kind of genetic test result, predictive, prognostic, or diagnostic, which in my opinion, is a much more serious issue than not having access to purely predictive genetic test results. But these are all single gene disorders. I mean, the question is, where has the genetic epidemiology taken us in the last 25 years? Well, basically, it's taken us beyond these single gene disorders and into a considerable level of complexity. It's called multifactorial disorders, which Peter is going to tell us a lot about this evening. It means that there are mutations in multiple genes, possibly quite large numbers of genes, each individually of small effect, but in combination possibly of a considerable effect, and also interactions between genes and other genes, and between genes and the environment. So you can see this is a completely different world from the world of the single gene disorders. It's also evident that we shouldn't think of this multifactorial genetic world as purely in terms of deleterious gene mutations, because if we added up all the multifactorial genetics, then what we would get is the general population. So they can't all be bad. They must average out to some good and some bad. So that also is a significant difference. But the complexity leads to some very difficult questions. There are questions of discovery of associations between genetic loci and an effect on what's called the phenotype, the physical organism. And what's being discovered is effects rather than causes, because, as Peter will explain, where this kind of research is not identifying the genes responsible for a condition in the way that the single gene tests did. There are quite often difficulties of replication. These are colossal experiments involving large numbers of individuals searching for relatively small effects. This is most definitely one of the branches of big science. And it's not always the case that an effect that is detected by one study can be replicated in other studies. But above all, there are difficult questions of interpretation, of what a discovery of an association means for in epidemiological terms, and ultimately in clinical terms, and therefore in actuarial terms as well. The real difficulty, once the science has been done, is perhaps in the interpretation. There's the interpretation of findings at a single genetic locus. Generally, at a single locus, there will be 2 variants of a gene. We have to assume that they both lead to viable organisms, or we would only observe one. And detecting these small effects is like looking for a very small signal among a whole lot of noise. So it's very difficult to do in the first place. And generally speaking, the effect is expressed as a relative risk. There's a low risk variant and there's a high risk variant, and you've got a relative risk of the high risk relative to the lower risk. And then even as difficult, if not more difficult, there's the interpretation of the results at multiple locations where you believe that there is that a large number of variants are collectively having an effect on the phenotype, the condition of the disease that you're studying. Because how do they combine? It's generally assumed that relative risks can be combined multiplicatively. And there is some evidence for that. But when you think of the number of possibilities that arises, even with quite a modest number of genes, it's evident that the effects of combinations of gene variants has to be based on a model rather than on observation. What this leads to in combination, adding up of small effects, law of central limit theorem, if you remember that, it leads to usually leads to a normal distribution of risk, where at the low end you've got people with lower risk than the population. At the high end you've got people with higher risk than the population. In the middle you've got the population risk. What I said about they can't all be bad. When you aggregate all the risks, you must get the population that you observe. As an example of the importance of the model in calculating these combined indices of genetic risk, which you then use as a relative risk in the model, I'd refer to a study I did with colleagues at Heriot a few years ago, looking at breast cancer and the effects of these multifactorial disorders. At the time, there were about 7 variants in seven different locations had been identified as associated with breast cancer risk. That came nowhere close to explaining all the risk that was there to be explained. And if you scaled up the risk from the ones that were known, then that suggested that the model with about 147 genetic loci would account reasonably for the risk that had to be accounted for. Now, with 2 variants at each location, that means 3 possible genotypes at each location. So that means the total number of possible genotypes in the model of 3 to the power 147. And that's a very large number. On a pretty conservative basis, it's larger than the number of people who have ever lived on the planet Earth by a factor of at least 1,000 billion, 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 billion. So we're not going to say let's go out and get a sample of 100 people with this particular genotype. And so the model is an absolutely central feature of the interpretation of the results of these multifactorial studies. So back to the question I started with: how is this going to translate into insurers' risk and insurers' use or not using genetic test results? Well, I think the context is very important here. If you imagine a universe in which nothing but genetic information is relevant to the risk, and insurers are not allowed to use it, then yes, that would put insurers in a very difficult position. But that's not the universe we're in either 25 years ago or now. 25 years ago, we had a universe in which genetic information, such genetic information as was available, single gene tests, was not available to insurers, but everything else was: medical history, family history, and everything that insurers normally use in underwriting. And because of the rarity of these conditions, the impact was such that the industry could agree to the moratorium, and if there were any adverse selection costs, it could absorb them. I've never heard that any have been noticed. And the retention of the ability to use family history was far more important to the insurance industry at that time than being denied the use of genetic test results. And now we're looking at a universe in which genetic information at a much lower level of effective, of predictive power, if you like, is being added to the mix of all the other things that insurers currently are able to use in their underwriting and reserving. And if you think of the picture of the normal distribution of the risk, adding up large numbers of quite small relative risks, or multiplying them together, then yes, there might be one or two people on the planet who've got combinations of all the risky variants, if there's a few dozens or 100 or so of those, but not very many of them. The vast majority will be somewhere in the middle, as in any normal distribution. And the kind of risks that entails are of the magnitude that insurers have been used to coping with since life insurance existed. And so I find the prospect of the new genetic information, whether or not it's made available to underwriters, is very unlikely to seriously threaten the viability of the life insurance industry. A conclusion that was true 25 years ago, and although we now know a lot more, I think it's not changed. So I'm looking forward very much indeed to Peter's comments on the subject, and any light he may throw on the risks that insurers may find before them, whether or not they know what the genetic information the person themselves may know. Thank you.
Thank you, Angus. In introducing Angus, you may have recognised that Angus was quite unusual in having had a successful commercial career as an actuary before having a successful academic career as an actuary. That kind of CV is so unusual that I've got someone else on my right-hand side who has a very similar career path. So Peter Joshi started out life as a commercial actuary, reaching executive level at organisations like Standard Life and Tesco Bank, before returning to science and academia in, I think, 2011. And Peter is now the AXA Research Fellow at Edinburgh University. His current principal research interest is the genomic basis for human lifespan, where analysis of the survivorship of approximately 1 million UK biobank parents is leading to fresh insights into how genetics influences longevity and how we might predict and perhaps shape healthy ageing. And such is the interest in Peter's work that I think each time he publishes new findings, it's covered not only in journals like Nature through to Radio 4, but also in The Sun and Metro magazine. So I'm hoping we'll find out what the fuss is about just now. And if I can raise the challenge to you, I did listen to Peter on talk radio last week, and I'm hoping that we'll get some more incisive questioning from people in the audience today than was obvious on talk radio last week. So there's a wee challenge for you. So very warm welcome to Peter Joshi.
Thank you, John, and thank you, Angus, for a very good warm-up. Thank you in particular for introducing the word multifactorial disease, which I want you to remember. Multifactorial means more than one factor, in particular genes and the environment here. And secondly, for introducing the idea of lots of genes of small effect. Okay, so aging is better than the alternative. And that's why I'm so often exasperated by us as actuaries and by society as general about the complaints about our ageing population. Increasing longevity is a sign of great progress in our society. But at the same time, I do understand we are concerned about the pressures on our funding systems and the perhaps bigger question as to whether all of the increase in lifespan that we're observing is an increase in healthy lifespan. My research program is about aging. I am interested in measuring aging, understanding aging, forecasting aging, and in due course developing therapies that will allow us to influence aging in the way that I hope will lead us to longer, more productive, healthier, and most of all, happier lives. In recent years, my research is focused on genetics and genomics. I think many of you are familiar with the idea that correlation is not equal to causation. Genetics, for a variety of technical reasons, is an exception to that. And when I identify a genetic association between a region of DNA and lifespan, I am able more confidently to say that I've identified a causal biological pathway than is the norm. And that means that genetics is a particularly good instrument for understanding the causal pathways underpinning aging. Okay, so what have I found? Well, we're beginning to reveal, but only beginning, I have to admit, how and how much DNA affects lifespan. I'll show you how DNA can already be used to make meaningful life predictions at birth, at least at a group level, but of a level I think that would be of interest in principle to actuaries. And finally, I'm going to look at the wider genomic revolution and talk to you about some of the real opportunities that that's presenting us, but at the same time, some deep ethical challenges. And some of those are indeed actuarial. Deoxyribonucleic acid. If you say it loud enough. No, that was something else. Actuaries are not the only people, though, who love acronyms. This one, though, I think you're all familiar with, DNA. So what is DNA? I think most of you will have heard that DNA is a double helix. And here the double helix is pictured in this cartoon representation. The really important thing to realize here is the two helices are mirror images of each other. Mirror images in terms of that helix running around the outside, and mirror images in terms of these bonds between them that hold the two helices together. Mirror images in that context in particular, can I get the slide? Yes, pointer here. If you see a green symbol here, you'll see a blue one here, and that's true all the way along your DNA. Okay, so it's a mirror image all the way across. And more importantly, it is these colored bonds, as I've colored them, that are your DNA code. That DNA code is 3 billion bonds long, 3 billion letters long, 3 million codons long, whatever term you want to use for it. You've got 2 copies of that code, one you inherited from your mother and one you inherited from your father. If you were to stretch the molecules end to end from one cell, it would stretch 1.5 meters. There are 30 to 40 trillion cells in your body, all of which have a copy of this molecule. And so the key thing about DNA is it's copying from cell to cell. And it's that mirror image property that allows that copying to take place reliably. It's a bit of a loose metaphor, but if you think of the easiest way to make a copy of yourself, it's to look in the mirror. And that's part of the way that DNA manages to do the copying as well. So if that's our DNA code, like any code, the issue is how is it decoded? Well, it's decoded in a part of a cell called our ribosome. The DNA can literally be thought of as being fed into the ribosome in much the way that ticker tape might have been fed into an old-fashioned computer as computer code. The ribosome then translates that code into instructions to assemble amino acids that you may have heard of. And those amino acids are then assembled into long chains in a specific order determined by the code. And those proteins are what goes up to make your cells and tissues, and differences in those proteins are what goes to make up differences amongst us. Okay, so DNA is a code that's copied again and again and again, and the proteins that are assembled using that code make our tissues and make us who we are. So, the first question I want to address with you is lifespan genetic? And I hope I'll show to you how that's about, does DNA determine lifespan? The historical starting point for this question is quite different from DNA. It's familial resemblance. I'm sure you've all observed how things run in families. You'll see that in 1886 here, Francis Galton published a paper where he observed that very tall parents had taller children than average. He also actually observed that very tall parents had children who were not quite as tall as themselves on average. With some wonderful Victorian language, he talked about the height regressing to the level of mediocrity, and that's where the term regression actually came from. So anybody who's performed a linear regression, this is what was meant when it was first used. But the key point here is to think height runs in families, taller parents tend to have taller children, and you can do some sort of conventional regression. The same is true of lifespan. Here's a graph of mother lifespan, and then that's been grouped, and the average lifespan of daughters amongst those groups is shown, okay? What you can see here is mothers who are living to 90 to 100 are having daughters who are living in the range of about 82 years. And conversely, shorter-lived mothers are having daughters who are living perhaps 75 years. Okay? So, big difference in the mother lifespans, smaller differences in the expected lifespan of the children, but the same process as Galton observed for height. And that's true not just for mothers and daughters, but all of the other relationships you might think of as parent-offspring. And it was true in a study we did of 3,000 people in Fife, and a study of 2,000 or so people we did in Orkney. So, lifespan runs in families, but how much? Well, if you are lucky enough to have long-lived parents, say your mother, perhaps she's lived 10 years longer than the average person in the population. How much might you expect to live longer? Well, 3/4 of a year. So not a great deal. I think many people who I talk to informally about this expect a bigger relationship. But that's a general relationship across society that we would see. And the same is true fathers. And if both had lived about 10 years longer than average, you might expect 1.5 years increase for yourself. Interestingly, and for the skeptics out there, importantly, we observe a similar correlation among spouses, and we're not expecting them to share unusual amounts of DNA. We do expect them to share environment, and so the question is the correlation between mother and daughter an environmental correlation or a DNA correlation? Another way of addressing that question is a term that geneticists call heritability. Heritability measures the proportion of the variation that's due to your genes. If the proportion is 0, I'm saying that that particular trait would all be about chance and environment. If, on the other hand, it was 1, it would be a purely genetic trait. However, heritability varies by trait, and heritability estimation, for reasons I've already begun to allude to, is tricky and controversial. There's always been a great deal of controversy between social scientists and biological scientists about nature and nurture, and that's at the heart of this estimate. So, what do geneticists think about the heritability of lifespan? Well, you can see here that we've got numbers ranging between 7% and 26%. If I was to, and you can see that the more recent numbers have tended to be lower. If I was to come up with a single estimate, I would say about a 1/12 or 1/8 of lifespan is genetically determined, and the rest of it's determined by the environment, and I think chance itself plays a particularly large part. So how does the heritability of lifespan compare with other things? Well, height. Galton chose a good trait to start with. Whilst there's some uncertainty about the heritability of height, it is much more heritable than many other traits. And you've probably seen that amongst people around you in your family. Things like BMI, somewhat heritable. Education level as well. I think we're confident there's a genetic basis to that. It remains very controversial whether or not that is 17% or 43% or somewhere in between. And then lifespan less so again. But still confident there is some basis in genetics, which I hope I'll illustrate more clearly in a moment. Okay, so does DNA determine lifespan? I've not really mentioned DNA in that piece of the talk where I talked about heritability of lifespan. With all of our modern molecular techniques, what can we say about lifespan and DNA? Well, give me a brief digression in a way. Imagine this is your DNA code at one particular part of that 3 billion long letters code that went in to make you. What does it mean? To be honest, I can't tell you. I can tell you a few things looking at this, but really I can't tell you very much about what this is doing. Some of my colleagues do try to feed codes like this into cells and understand what happens within the cells when that happens. Even then, if you do understand what's going on at a cellular level, it's very difficult to take that up a level to a tissue and so on to an organismal level, and then again to a trait outcome like lifespan. So it's very difficult to look at that code and say what it does. An alternative would be to think, well, what happens if we change one letter in this code? You might think of that in a computer program. What happens if we change one line of the computer program? What does that do? Run the computer program again and see what happens, or maybe run it thousands of times with and without the change and see what happens. That would tell you something about it. I think you'll be glad to hear that it's considered highly unethical to change one of your letters of DNA and run you again thousands and thousands of times. So that's not what we do as geneticists. But we're in the very fortunate position that nature provides us with a natural experiment of that form. Nature occasionally changes one of these letters. And here's a wonderful second acronym for you, SNPs, or SNPs in our world, not anything to do with Scottish politics, but single nucleotide polymorphisms. Occasionally across the genome, there is variation in the letters. So this might have been your letters at this particular part of the genome. The person sitting beside you might have these distinct letters. If we were to analyze their traits versus your traits, we wouldn't be able to come to that many conclusions. Different environments, so many different letters when we think about all of the letters across the genome. But if we analyze enough people with enough precision, we can begin to understand what one of these single letters might be doing to that trait outcome. And that's indeed what we do. So this is a picture of the genome. These are chromosomes. The first chromosome is the biggest one, and then you can see all of the chromosomes pictured here. So what do we know with all of the effort that's been put in about how these chromosomes and each part of these chromosomes affect things? Well, each of these spots represents a specific association between one region of the genome. Sorry, the genome is the collection of your DNA. I don't think I said that already. One region of the genome that has been associated with one particular disease, for example, digestive disease, cardiovascular disease, and so on and so on. So we've discovered in the past 15 years thousands of robust associations between specific parts of your DNA and disease outcomes. What do we know about lifespan? When I started my work in 2014, this was the only known robust association between lifespan and DNA. A huge contrast. And the reason for that contrast is partly that the heritability, the genetic basis of lifespan is quite limited. There's so many other factors. And partly the small effects that Angus talked about earlier. That result can also be pictured like this, and I'll dwell on this slide for a moment because we're going to return to slides like this. Along the x-axis now is your genome. So we see each of the different chromosomes represented here along the x-axis. And every one of these spots is a region of the genome that's being tested for association with lifespan. And then on the y-axis, we're seeing the strengths of the association. So I hope it's obvious to you that there is one strong association or one region of strong association that's been highlighted in this graph. In fact, as a genomicist, this line here is very important. That's the line that says above that level, we believe we have a reasonably robust association, and below that, we do not. We don't know if the apparent association is just chance. Or it might be signal. And you can see here suggestions of signal, but you also need to be concerned that it might be just noise and chance. Okay. So What did those people do to do that study? What they did was they found 10,000 particularly long-lived people. These people had lived beyond age 90, in fact, in that case, and compared them. They called those cases. You may have heard of case control studies in epidemiology. They called those cases, and then they controlled people who had more normal lifespans, or were still alive, in fact, in both cases. Can see that that worked to some extent. They found one genomic region and a robust association that has now been replicated. However, it's very expensive to do. You have to go out and find 10,000 people that are aged. You need to take their DNA, having got informed consent from them. We've done something else. We've said, let's study lifespan, but because when we recruit people, they're alive, we're going to have to wait a long time. So let's instead ask them about how long their parents lived. And we're able to make precise inferences from your DNA of what your parents' DNA is. So if your parents particularly long-lived and you have a certain kind of DNA, we can make some admittedly probabilistic inferences about what was going on with their DNA. And so here you can see symbolically that represented. I'd like to thank my PhD student, Paul Timmers, who did most of the analysis that I'm about to present, and in particular, gathered together these 1 million lifespans that allowed us to do this analysis of small effects with statistical robustness. And what did he find? Well, we found, I hope you now have some sense of this graph. Things that rise high on this graph are robust associations. And then we found 12 robust associations between regions of DNA and lifespan. Okay, I'm not expecting you necessarily to recognize these gene names or genomic positions, but I think the first thing I would ask you to do is have a look at the column years in this table. And what you can see is that typically the effect of carrying one genetic variant relative to the other is about 0.3 to 0.5 years. So small effects, if you think about the standard deviation of lifespan being, say, 10 years, and the total variation of lifespan obviously being much greater than that. Look at the frequencies. The frequency of the beneficial variant and the deleterious variant are, broadly speaking, between 10 and 90%, so both are reasonably common. There are statistical reasons for that. I can't find incredibly rare things. If it only exists once in a population, it's very difficult for me to do statistical inference on it. And similarly, that's the reason why I've not found smaller things. I need big enough effects, even if they're small, to find. And finally, have a look at the diseases that so having found these variants, we asked ourselves, what diseases did these variants also associate with? And you can see a very strong theme of cardiometabolic disease here, as well as some other things. We then did something that is a little bit complicated, but instead of looking at phenotypic correlations, we know the underlyings of genetics of lifespan now, and we also know the underlying basis of heart disease, and we're able to, genetic basis of heart disease, and we're able to look at how the functions of those genes correlate, okay? And we get something out of that is a genetic correlation. It's got all of the properties you would expect of a correlation, but it only relates to the genetic effects. And here you can see the genetic correlations between survival, longevity, and various different traits. Let's start with heart disease over here. We can see that there's a fairly strong negative genetic correlation between susceptibility to heart disease and how long we live. And I guess I've privileged that in the previous slide. There's also a strong negative genetic correlation to propensity to smoke and how long you live. Again, no great surprise. I won't go through all the traits. You will be interested to see there's a positive genetic correlation between the genetic trait, the DNA influence on how long you live, and the DNA influence on how much education you receive or how long you stay in education strictly, and survival. And you'll see one, you may be even more surprised to hear, there's a genetic trait, happiness. There are DNA variants that influence how happy you are. And there's a genetic correlation between that and lifespan. Before you feel too smug, there's actually a negative genetic correlation between educational attainment and happiness. Okay, so we then turned that question on its head and said, okay, let's go out there. We've seen major disease categories have been associated with genetic variation that we know affects lifespan. Let's now look at disease categories as categories and look at the genetic variants that affect diseases and see if they affect lifespan. So for Alzheimer's disease, there's a particularly strong effect variant, the ApoE variant, that is also protective of lifespan. So protection against Alzheimer's disease by this variant is also protective against lifespan. We tested, you probably can't see it, a whole lot of other variants, and we saw what looks more like noise to me, but certainly weak associations between these other variants that affect Alzheimer's disease, admittedly much less than the ApoE variant, and lifespan. We then went on and looked at the genetic propensity to smoke and the associated suffering of lung cancer. And here we see perhaps what you might expect. A variety of genetic variants are known to affect smoking and lung cancer, in this case reducing them, increasing lifespan. And then, again privileged, what we're seeing here is a whole lot of genetic variants that we know influence susceptibility to cardiovascular disease. Each of those, if they're reducing susceptibility to cardiovascular disease and are coming out above this line in black, and you can see lots and lots of cardiovascular disease variants affecting how long we live. And then I think more surprisingly, we looked at, there's a lot of genetic variants that are known to affect susceptibility to cancer, and we're seeing a much less clear line of sight between genetic susceptibility to cancer amongst these variants and how long we live. That's probably because the effect of the genetic variants on cancer may be smaller, and the effect of cancer on lifespan may be less than cardiovascular disease, or at least it may act only on one pathway. What we also see that's quite interesting is the same gene here. This gene, a susceptibility to cancer, is associated with a longer life. Okay, so and that's happening because this gene is associated with a susceptibility to cancer, but a protection against cardiovascular disease that's offsetting it. Okay, let's not dwell too long on this slide, but by getting all of that information across the whole genome, I'm able to then look at gene pathways and biological pathways, and what we're seeing is the most significant associations associate with lipid metabolism, so your cardiovascular lipids, and with your brain function. So it looks like heart and brain genetics are influencing how long you live. Here we see that those effects vary by the stage of your life and by your sex. The Alzheimer's variant affects women more and later in life. Conversely, this heart disease variant affects men more and early in life. So, I hope I've persuaded you that DNA does influence lifespan. The next question, I guess, of particular natural interest in actuary is, can it actually predict lifespan? Before we turn to that, and thank you again, Angus, for the excellent introduction, it's worth thinking about the framework that we've been in for a long time, as Angus highlights, 25 years or more, traditional genetic risk prediction. And this is an extract from the NHS website that I've paraphrased. Inheriting BRCA1 genes, faulty BRCA1 genes, raises the genetic risk of cancer. Those variants may be passed to your children, and indeed you may well have inherited from your parents if you carry them. But it's far from only the cancer risk. There's a myriad of other genetic and environmental causes of cancer risk. And then the key point here is speak to your GP if cancer runs in your family. If you don't have cancer within your family to any great extent and go to your GP tomorrow and say, oh, I was at a talk last night, I want some genetic testing for cancer, he's just going to turn you away. It's only if there's a significant amount of cancer running in your family that you would be sent to see a genetic counsellor who would then look at actually the way it's arranged within your family. And if it's mainly your wife's family, he's going to say, well, it's not relevant to you, and so on and so on. But if it does run in your family, he may recommend you for a genetic test. That test could be inconclusive. It could cause you anxiety to no purpose. It could lead to action. So Angelina Jolie's genetic counsellor obviously discussed action because she was found to carry the fault of BRCA1 gene and had a mastectomy, as I'm sure many of you are aware. The genetic counsellor may also advise you about telling your family and also about family planning. And not all the time, but there is a possibility, at least in the context of IVF, of pre-implantation genetic diagnosis of the embryos and only the risk-free embryos being intended to be implanted. So that's current technology. That's existed for quite a long time, okay? So what's changed? Well, sorry, forgot about this slide. This slide basically says the same thing, but it highlights the fact over here on the right that 99.9% or 99% of the population might have the low-risk variant, which is why your genetic counsellor might be unwilling, your GP might be unwilling to send you to see a genetic counsellor unless you've got some evidence beyond having been to this talk tonight. Okay, so what's changed? Now I've hopefully shown you that I have a whole genome basis of understanding of the genetic basis of human lifespan. I have these particularly strong signals, but I have a mixture of signal and noise here, and I can use some machine learning techniques to combine all of that, and perhaps taking your DNA, reading it, combining all of that, and come up with a personalized score for you that takes all that knowledge together. And as Angus told you earlier, that personalized score will typically have a very much close to a Gaussian distribution, essentially because of the central limit theorem. That's been done relatively recently in a paper that was published about 6 months ago for individual diseases. The people did exactly what we just described. They worked out a score, a polygenic risk score, because it's looking at these many genes, for here, first of all, illustrated atrial fibrillation. Okay? And what they've done on the x-axis here is the percentile of the polygenic score for atrial fibrillation. That score could have been made at birth for these people. You would have just needed to take a cheek swab and run through the algorithm I just described. There's no other information being introduced into this. No information about socioeconomic status, parents, family history, anything. Just a cheek swab of your DNA at birth. And what they found was that people that were in the very top few percentiles, or the safest few percentiles at least, might have 0.5%, whereas people in risk of atrial fibrillation, whereas conversely, a 4% risk for the people at the other end of this percentile spectrum. So an 8-fold increase in risk. If we're thinking about critical illness policies, that's clearly far from immaterial. Similar results for type 2 diabetes, for inflammatory bowel disease, and for breast cancer. Okay? So cheek swab at birth, very distinct risks of these diseases. So what about lifespan? Well, we did something a little bit different, but in essence very similar. Polygenic score, just as described, cheek swab at birth potentially, or at any point in life, and we can score people just based on their DNA into deciles. I hope I don't have to explain what a bottom and top decile is for you. Okay, and so what did we find in terms of the survival differences? Well, here are the survival curves for parents in the top decile versus the bottom decile, men and women, mothers and fathers, obviously, in this context. And the median contrast that we find in survival time between the top and bottom decile for the score is about 5 years. So I'd say that's not immaterial in actuarial terms. If you're underwriting a set of annuities and you've got a 5-year difference in expectation of life between one identifiable pool and another, well, you'll tell me later whether you think it's immaterial. And we saw that as well, not just in the parents, but in the subjects. We're able to do this better in Estonia because of the structure of their sample. Smaller effects in Estonia for 2 or 3 reasons we might discuss later. And that's illustrated further across all of the different combinations in these graphs. You can see it's worked in every case. I'll maybe skip this slide in the interest of time. So I thought I'd now step back from that and talk about genomics more generally in the news. There's hardly a day goes by, I think, certainly not a week goes by when there isn't some announcement of some new genetic or genomic research and its potential impact in our society. The context of that is really this slide, I would argue. This slide is illustrating the cost of getting DNA information from us. In 2001, the cost of sequencing your whole genome was $100 million. It's now $1,000. That, on this graph, which has got a log scale, you'll see Moore's law illustrated. And we think about the progress we've seen in electronic technology over that time because of the effects of Moore's law. Well, the genomic technology has, albeit from a much higher base, arguably, proceeded much, much faster. And we're now in the situation where potentially, I think, every baby that's born in a rich hospital in America may see their genome sequenced at birth. Not quite yet, but I think it's coming. I've already talked about this paper, but the polygenic risk score paper that looked at the relative risk of atrial fibrillation and breast cancer and so on, but it's attracting a great deal of public interest. I'm sure many of you heard of a different situation where we saw a Chinese scientist editing the genome of someone. So this, what I do has got nothing to do with gene editing. I just measure your genes as they are. He edited the genes of an embryo and then implanted them. And that's been extremely controversial. It's ethically controversial. It was doubly controversial because there was no real ethical consultation about carrying out this experiment in advance of it happening. And then I think you will also have heard of gene therapies, the hope that we can inject different genetic material into ourselves, not passing on to our offspring, but that genetic material might compensate for a fault in our own genetic material, or might actually act within our cells in a way that, even if we don't have faulty genetic material, might improve outcomes for ourselves. So, the world is changing. In the past, genetic analysis was restricted to very large effects within affected families. Individual genetic risk for death or common disease was simply not possible. It was possible for, as Angus described, very rare diseases that had very large effects like Huntington's disease. And that situation meant there could be a very successful ABM moratorium on insurers using genetic testing. And I think the key to that was the fact that family history and other medical procedures were still disclosed to the insurers. And we have had screening during IVF for rare, large effect familial diseases. Okay? Possible future, as I've described already, everyone might be sequenced at birth. That would mean the day after your baby's born that we could calculate polygenic risk scores for everything: educational attainment, longevity, risk of any given disease, happiness. That process is also, though, likely to throw lots of variants of unknown significance up. You carry about 300 DNA differences that are not present in either of your parents. It's very difficult for me often to say what those DNA differences will do because they're often unique to you. Psychological impact of these scores, of the susceptibility to known diseases, or these variants of unknown significance, many of which, the vast majority of which, seem to have no effect. And the potential, depending on the way the regulation develops, for a deep insurer-customer asymmetry in information. Potential, and sorry, in terms of anything I've described, and certainly what I'm about to describe, I'm not advocating these things, but this is a possible world that we might end up in, and unless society acts, we may end up with it without choosing. There's possibility in principle to screen for IVF for any of these genetic risk scores. Gene editing we've already seen happen. The really controversial thing about that is the children that were born with these gene edits will pass those gene edits on down the generations. And potential for gene therapies for many major diseases, perhaps changing the outlook for longevity substantially. Okay, I must thank a very large group of people. Firstly, the people who've been kind enough to fund me to do this research, the Medical Research Fund, the AXA Research Fund, and the University of Edinburgh in particular. The people who've provided the data, whether that's the scientists that have gathered the data or the participants that have actually participated in the studies, and in particular, all of those involved in UK Biobank, which has been the bulk of the data that I've been able to analyse. And last but no means least, my colleagues pictured here at the University of Edinburgh and a rather sunnier Lausanne. Okay, so in summary, we're beginning to understand direct lines of sight between your DNA and how long you live. The effects are small, though, at any particular site. However, combining all of that information, we can now make, at least at a group level, meaningful distinctions in expectations of life, and in principle, those could be distinctions could be drawn at birth. And then finally, the genomic revolution is presenting real opportunities and some deep ethical challenges to our societies. Some, perhaps not the greatest, of those are actuarial. And I would suggest to you that technology is currently developing faster than policy. Thank you.
Thank you, Peter. That was fascinating and thought-provoking, and I'm sure, as we're about to find out, question-provoking as well. Before I invite questions, can I just remind those on the live stream that if you'd like to contribute a question, you can do so via Twitter using the hashtag IFOAlecture. I see we've had one Twitterer already, but please, others, contribute via the Twitter feed. To everyone, those online and those in the audience, there's a prize available for the best question from a young actuary, so anyone within 15 years of qualification will be eligible for a prize. And I'm told, hot off the press, that Alan Watson is no longer eligible, so the field is open. So can I maybe ask, first of all, for a show of hands in the room for those who would like to ask a question? Nothing so far. Well, one up at the back, so thank you.
Hello, Adam Murray. So early in the talk, you talked about these genetic factors being quite small. I was wondering whether this is maybe just an effect of the fact there's a lot of things you can do to kill yourself, and possibly if society was safer, maybe it would be more of an effect.
I think that's right. If we certainly, susceptibility to death from buses would seem likely not to be at all genetic. Well, maybe. And so I think that we talk about the proportion that's due to genes and environment. The very simple model you might think is that your lifespan is your genes plus environment plus chance, and the proportion would there move towards genetics if we remove chance. Or make the environment more homogeneous. If we all grew up in identical environments, had identical diets, but different genetics, then you would expect an increase.
Would we expect something like that to happen?
I think I'll leave each of you to form your own conclusions as to the sort of future of our society and its homogeneity or heterogeneity. I think what we do expect to happen is more and more therapies to come online. Whether or not that actually changes the balance that I've just discussed, I'm not sure.
Thank you.
Thank you. I'll maybe take the opportunity to ask a question, Peter. I know I've heard you talk before that if our DNA at birth does say anything about the probability of our age at death, it may actually be that some of those factors are already observable later in life, either through BMI or cardiovascular disease. So what's the current thinking on how much additional information DNA gives beyond those observable facts?
It's a very good question, John, and one to which I don't think anyone, certainly I at the moment, don't have a particularly good answer. What John's saying is that if your genetic propensity to live long is about cardiovascular disease, and we can measure your BMI at, say, 50, we can measure your cholesterol level, we can give you an ECG, is the genetics going to tell us anything more, or has the genetics already had its effect and we're just waiting for that to emerge later? I think that's a very plausible model. It's certainly one of the next pieces of research that we plan to do. We have seen suggestions that in the context of cardiovascular disease, that the clinical factors already explain a great deal of the variation, and the genetic factors do not necessarily add that much more. Having said that, as we get bigger and bigger studies, our understanding of the genetics is getting better and better, and so we expect there to be a shift towards the explanatory power of genetics even in this context. But how big that shift will be, I think, is yet to become clear.
Professor Cairns at the front row.
Andrew Cairns. For the data set, the biobank data, does that also include some socioeconomic information? So in your results, you had a sort of 5-year range between the different deciles. So if you also included some additional socioeconomic information, would that allow you to widen that, and if so, by how much?
Yes. I think a number of members of this audience are actually probably more familiar with socioeconomic underwriting and longevity than I am. Yes, the data set does allow to do that. We actually, because we're very focused on the DNA, treat the socioeconomic status as a noise variable in our analysis. So we do include it, but I haven't separately analysed its explanatory power as opposed to the DNA.
Hi, Colin Ledley here. I was wondering if, Peter, you had views on how insurers' underwriting practices will or should change, and whether the ABI moratorium will or should be abandoned in future.
I think this is very difficult. I think there will be a great deal of hostility in society, and particularly, I think, amongst clinicians, to the use of genetic information in underwriting. I think I can see all sides of this coin. We as underwriters are kind of used to bring into account all sorts of factors, socioeconomic status. Well, it's not that easy to change your socioeconomic status, and yet we're allowed to use that as an underwriting factor. But people see their DNA as particularly sacrosanct information. There's a lot of concerns about personal privacy in DNA information, for example. So I think these graphs illustrate the potential utility, and I think more concerning to us, the potential customer insurer asymmetry in information if the customer knows this information and we don't. At the same time, I think it's going to be seen by society, and I think perhaps by many of us here, as undesirable to use that information that you have no control over, you're born with, in underwriting. And I think it's far from straightforward. But these graphs, I think, more than the graphs that I presented, where we're seeing modest difference in long-term survival chances at the moment, that may change, of course. But these graphs suggest to me that there is a storm brewing, and I'm not sure what the resolution is. There's a question. Peter Roberts.
Oh.
Peter.
Peter, microphone's on its way.
Microphone's.
Thank you. Peter Robertson. Having had some involvement back in '96, I'd maybe try and make a comment there and partly answer that and then ask you a question. In '96, there were 4 clear groups. There was the insurers, otherwise known as the bad guys. There were the customers, otherwise known as the poor unfortunates. There were the clinicians trying to help them, and the argument was, remember, it was single gene stuff. If we can't test these people, we can't treat them. And there is no point in wasting our money, as Angus said earlier, on treating people who don't have the condition. Then there was the 4th group, which was the commentators. who were very definitely on the side of the clinicians and the poor unfortunates. And the insurance industry was really making its life difficult for no good reason. If you take that forward to now, those four groups have been joined by a fifth, and that is the people who produce the genetic tests, because then it was only the clinicians who were doing them. Now there is an industry designed to produce genetic tests, very often producing information of complete, totally useless, like you are, 3.6% North American Indian and 22.5% picked or whatever it is. And it's like, how did you get that? No idea. So you've got lots of genetic tests now that have nothing to do with treating people. But I think, and this comes back to Colin's question and to your response to him, the journalists, the commentators still think, as they did in '96, there is a killer gene for cancer. There is a cancer gene out there, even though everything tells you there isn't. And so until you can get over that, anyone who uses that information is always going to be the bad guy. Sorry, I think I ended up losing my question in that. Apologies.
No, I agree with what you're saying, Peter. The other distinction that I think is implicit in what you said is that the family history question, and the clinicians were only, as I sort of highlighted earlier, talking to people with a family history of these diseases. That's not the case now, or at least wouldn't be the case in the scenario that we're discussing.
Thank you. I'm going to break the monopoly of the room in Edinburgh because we do have some questions via Twitter just now. So first one, Peter's from Nicola Higgins, who asks, are you aware of any studies on epigenetics and the impact on longevity? And if so, what impacts have been seen there?
Right, so, epigenetics. This DNA molecule that we're seeing here is more complicated than I made out, inevitably. Along the outside of this is a backbone of molecules that give rise to what are called epigenetic effects. Epi meaning above genetics. And in essence, what those molecules do, amongst lots of other biological processes in our body, they switch on or off these genes. Okay? So the majority of epigenetic process is, for example, the one that how do we decide when I'm a cell in your body, whether I'm going to be an eye cell or a brain cell or a heart cell. It's epigenetics. I've just said to you, every cell in your body is the same DNA. Well, how does it decide what to be? It's epigenetic, and it's because the cells code themselves with these epigenetic effects. So epigenetics is fundamental to biology. What we know is that, for example, if you have a particularly high diet and high BMI, your epigenetics change. So the way your cells behave change, and that's probably no great surprise to you. I think the question can be split perhaps into two parts. The first is, would epigenetic changes in measuring epigenetics that differ across every cell in your body in itself be prognostic of something? And I think the short answer to that is yes. We know that if we measure epigenetics, we can construct something that's called a clock, and we can measure your age because these effects change over your lifetime. And we actually know that it can infer biological age beyond chronological age, and that's in turn prognostic of lifespan. So yes, epigenetic effects exist. Now there's bigger questions that I think there's much less consensus and there's much more coverage in the press about these effects than I think the vast majority of geneticists are comfortable with. And that's whether or not these epigenetic effects cascade down generations. The conventional wisdom, and the wisdom I would run with, is that broadly speaking, epigenetic effects are not passed from generation to generation. And so we couldn't do a test at birth of these things. It's as you age, as you consume a certain diet, as your cells differentiate, we get these epigenetic effects. And they might be one way of measuring how old you are, in a way that DNA is not. I could take your DNA, broadly speaking, it's not quite as simple as this, at birth and now, and it's the same. Your epigenetics is evolving all the time within your body.
Thank you, Peter. I'll go for one more from Twitter from Stephen Baxter. With dementia a growing cause of death, can Peter offer some thoughts on the likely genetic hereditariness of dementia? And would we expect the explanatory power of genetics to rise or fall with the rising prominence of dementia?
So, I actually can't remember the heritability of Alzheimer's disease. I suspect it's in the range of about 30% because most of these traits are in that sort of range. Part of that non-heritability is that it doesn't affect anybody who lives to age 70. So in terms of outcome and lifespan, it's not going to really affect us. So it doesn't affect anybody. It affects a much smaller proportion of people in that range. As we live older, I expect susceptibility to Alzheimer's disease to play, and potentially, hopefully, therapies for Alzheimer's disease to be a much bigger part in how long we live. I'm not sure if that overall means our genes will affect things more or less, because if we're suffering from less heart disease, which was heavily genetic, and more Alzheimer's disease, which is also genetic, it's going to be a trade-off.
Thank you. I think we've got time for one or two more questions in the room in Edinburgh, please. Any arms being raised? No. I'll just quickly check so I haven't missed anyone else on Twitter. No, I think that's probably last call. Going.
I guess I maybe would have a question for you, and that is whether or not a 5-year distinction in lifespan between the top and the bottom decile is meaningful in underwriting terms, either for annuities or indeed in terms of life insurance underwriting at earlier ages in terms of the implied hazard ratio, and in terms of the underwriting practices you're seeing at the moment.
Peter, Alan Ray. I think I'd say it definitely is. The question is, if we were looking at pensioners or deferred pensioners, how much of that 5-year can we already infer from socioeconomic and others? So, if it was 5 new years of differentiation, it would be hugely valuable to be able to let's say at a pension scheme, if you're doing a bulk buyout for $1 billion of pension liabilities, to be able to swab all the pensioners. in that kind of world, but yeah.
Any other views on Peter's question? Yep.
It's Andrew Cairns. So on this same point, I mean, I think the issue probably relates to the sorts of work that Angus has done. It's about at what point do people actually have that information about where within that spectrum do they lie? And at the moment, probably the vast majority of the population don't have that data, so it's not immediately of consequence, but it's really in 10, 20 years' time, if everybody gets this swab at birth, etc., and has their prognosis, then what do they do with that? And then is there a significant selection effect? And I think that's where at least we've got the benefit of time to be able to think about that and discuss what the consequences are. But I think now maybe not such a big effect, but in the long run it could be. Yeah.
Thank you, Andrew. I think we've got a comment as well from Angus MacDonald now.
Yes, following on from what Andrew said, I imagined in my remarks a universe in which genetic information was the only available information and nothing else, and that's precisely the scenario that Peter invented of the swab, everybody having a swab birth and having their whole genome sequenced and then learning nothing else about them at all until they come to buy a pension at age 65. So it's very much a case of what does genetics add to the information, the conventional information that we have just now at any point in the insurance purchasing process.
That's great. Thank you, Angus, and thank you again, Peter, for taking those questions. I'd like now to introduce our closer, a very well-kent face to the Edinburgh audience. Alan Watson is a consulting actuary with Aon in Edinburgh. He advises clients on all aspects of pensions and has several scheme actuary appointments. He's also a member of the IFOA's Council Management Board and is leader of the Scottish Board. So, very warm welcome to Alan.
We've had a fascinating discussion tonight. I wonder how many people wondered what we were going to get tonight, and I've actually found it very enlightening. We started off with Angus, who introduced us to genetics and actuarial work and gave some of the background to insurers and genetic testing. I think that was a helpful way of doing things to give us a bit more introduction at the start before we launch into Peter. But Angus reminded us that there are positives as well as negatives from all this aspect. And he outlined many of the difficulties of using genetic information and reminded us that other information was available and it's often much better than the genetic information. Peter then took us through some of his research. I did read the paper that's online. And I'm glad you told us what a lot of these things were, because I had to go Google quite a lot of the terminology. But you reminded us that, yes, increasing longevity is good news. We do worry as if this is a bad thing, but people living longer shouldn't be seen as a bad thing. And he took us through a high-level explanation of DNA and genetics, which I hope you all found as useful as I did. And few of us would doubt that lifespan would run in families. But I think we probably were quite surprised that maybe only a 1/12 of the influence is Peter's opinion. And there are other much more influencing factors, and lots of them, is basically what he was telling us. And there's a lot of really small effects. The disconnect is the sort of DNA traits of happiness. We discovered this DNA traits of happiness that I was fascinated at. But how happy is the average actuary was something he did raise, because it seems to be you're not happy if you're educated. But he was combining these factors is one of the important things. Is what he was doing is, rather than looking at individual bits of DNA, was to try and combine what's the whole information we're getting from this, and to come up with that 5-year difference in life expectancy between the top and bottom deciles. I think we all, that sounds material. Alan Ray confirmed he thought that sounded material, but then we discovered later on, as I'll talk about, maybe not quite as much. But we are near this point of the genome sequencing at birth, as you described, and that poses lots of questions. And I'm glad you've started to pose these questions. I hope other people are posing these questions, because these questions need to be considered now. And I think that's one of the things that you're raising, is that we are getting to that point, and the challenge of technology ahead of policy is what you mentioned, and that's really important. We then got on to variety. We got some questions, which I was pleased to see. Adam Murray made a very valid observation about the other things that affect lifespan. John Taylor, chairman, coming in second, that's an improvement. We've got people to start saying things. He raised the query of where pure lifespan and other influences is coming in, because I think that's the other thing. We struggle on reading the paper. Where is the difference between pure lifespan influences and all these other influences? And I think you've shown that it's all mixed together. Andrew Cairns talked about the influences of socioeconomic factors, which raises important further questions. And Colin Ledley asked about the practical question of where does insurance underwriting actually go? I was pleased that we started to get into some of the practicalities of this as well. Peter Robertson, who said he was there in 1996, so I think he's ineligible for the Sloan Prize, I presume. And educating those involved in the subject is going to be one of the challenges. I think because there's lots of people that fear genetic information when it's probably not as fearful as we imagined, but it's educating people about that this is actually can be quite helpful. We then had 2 Twitter questions. So I'm going to predict they probably are going to be eligible for the Sloan Prize, because I doubt anybody qualified for 15 plus years has even heard of Twitter. But Nicola Higgins asked about epigenetics, which I was a bit worried that Peter was, but you then dealt with it and gave us a thorough explanation of it. And then Stephen Baxter came with the question of dementia, which is another, the challenge of explanatory power of genetics. And then, I don't think you're supposed to ask the audience questions or something like that, but you did pose that question that we talked about before. Is the 5-year gap meaningful? And I think most people will agree it is, but then it suddenly becomes a bit more difficult. Alan Ray said yes, but as we accept that the challenge is to differentiate it from other factors. And that's, as Angus said at the end, it's all very well to be swabbing everyone at birth, but in reality we're going to have all these other influences that are going to have equal, or far more influence. But in summary, I've found it fascinating. I'm glad you've taken it so much further than when you did a small session for us about 2 years ago, when you talked about a lot of these concepts. And I'm glad I actually understood some of it at the time, because it's helped me understand it a lot better tonight. And I'm hoping people will have found this really helpful and not too full of actuarial theory, which is always a good thing. So.
Thank you, Alan, not least for giving us an extra correlation tonight between Twitter usage and post-qualification experience. Thank you. I think it remains for me just a couple of closing points. So for anyone who asked a question tonight, if you've got any notes, please can you share them with the members of staff in the room tonight to help us type up the questions. I'd particularly like to thank our contributors from the table this evening. You know, Angus for a very an exceptionally well-informed opening introduction to Peter's talk, and Peter for taking us to the vanguard of research on genomics. And then, you know, Alan for rounding up towards the end. Thank you to all 3 of you. I've certainly found it a fascinating hour and a half, and I'm delighted to have survived an hour and a half as a Glaswegian. That's a feat in itself to have done that. Final vote of thanks just to the staff. These events require weeks and sometimes months of preparation to pull together and run smoothly. So thank you to the staff too for bringing this together. So we'll hopefully see you for the next sessional event. Keep your eye on the website for that one, and thank you all very much for coming along and contributing this evening. Thank you.